Hi, my name is Natalie Leger, and I'm going to be explaining synapsis of chromosomes during meiosis. Meiosis is a specialized form of cell division where gametes or sex cells are produced. So synapsis occurs during prophase 1 of meiosis, where homologous chromosomes form pairs. So synapsis is the alignment of these chromosomes, which facilitates the sharing of genetic material between non-sister homologous chromatids. So as you can see here, our two chromosomes have gotten together and overlapped their chromatids where they are sharing genetic material, and they are now parting on good terms. So our first pair of chromosomes are the pink and blue, which I have depicted using pipe cleaners. And we can see that in the loci present on these chromosomes, there's an inversion of the underlying loci I, J, and K. So this inversion is going to cause compensation, which we can see in the twisting or looping of the pink chromosome to create as much alignment between the loci as possible. So then we have a crossover between J and K. The centromeres are still present. Here's this crossover, and here are the gametes that are produced. So I have illustrated each chromatid that is created after the crossover. And you can see that the green and purple are both non-viable gametes. Why is that? That's because there are duplications of the loci, and also all of the loci are no longer present. So for the green gamete, we no longer have H and G, and for the purple, we no longer have L and M. So here are our second pair of chromosomes, and they're the same this time, so no compensation occurs. We have a crossover, and here both the loci and the centromeres are present this time, and this produces all viable gametes. Because there's no inversion and we just have completely homologous chromosomes and no differentiation of loci, the produced chromatids, they all have the exact same loci in the exact same pattern, and therefore the gametes are all the same and they're all viable with all loci present, no duplications, and they each have their own centromere. So these are my references. Thank you very much.